this video tutorial, we will understand the concept of blue-white screening. Okay. So, in the last video, we have seen that when we are inserting our gene of interest into the plasmid, there are three different possibilities. Okay. So, you can see over here that the first possibility is that the gene of interest will be in our leg Z site. Okay. That means the site is getting disrupted over here. Okay, so the next one will be outside of the leg Z site. Okay, and then the third category or the third case is that there is no insert over here. Okay, so we have done the gene cloning, but how we are going to identify that whether it has the colony that is produced has our gene of interest or not. Okay, so you can see that if the leg Z site is getting disrupted, that means our gene of interest is present over here, which is confirmed. So it will give white colonies when you are doing it on XGAL plus IPTG culture plate medium. You will get blue colonies, that means no, it is not inserted over here. So it is outside somewhere, and here again the blue colonies, that means the gene of interest is not present. Okay. Now let's again learn this with deeper example. So you can see over here that there is, okay, this is the bacterial plasmid. Okay, so it has this ampicillin resistance gene. Okay, then this is leg Z, that means lactose breakdown gene, where it is a multiple cloning site over here, restriction site, so human cell. So you know that first we need to isolate this plasmid DNA and we need to isolate this human DNA. Okay, so it is getting fragmented over here. So you can see over here that by using restriction enzymes we have done the cutting of this leg Z site. Okay, so you can see that there are different plasmids, I mean the same plasmids, so the number of copies are more of the same plasmid. We have used restriction enzymes on them. Okay, now we are with the help of DNA ligase, we are joining this. Okay, we are ligating this. Okay, so here the gene of interest is present over here. Okay, but here you know gene of interest is not present. Here again the gene of interest is not present. Okay, so now, okay, let's say if the site got disrupted over here. Let's say if the site got disrupted over here, here you can see that means the leg Z gene will not be fully functional. Okay, so when these plasmids, okay, are introduced into the host DNA, bacterial DNA, and if they are grown onto the Petri plate, so they will grow on the Petri plate. Definitely they are going to grow on the Petri plate. But how we will identify that this is only the gene of interest or not? So the colony that has hindered or interfered or a mutation in the leg Z gene will not show blue color. I am repeating, it will not show blue color. They will be of white color. Okay, so they have no leg Z gene, that means lactose broken down is not possible. So colony carrying recombinant plasmid with disrupted leg Z gene will be white in color. So this white is the recombinant one. Okay, students often get confused that the blue is recombinant one, but no, the blue part gene is getting disrupted because of the insertion of our DNA. And the blue one, they are the non-recombinant plasmid with the intact leg Z gene. Okay, so we have to select such colonies, the white colored colonies, for further processing. I am repeating that there will be three cases over here. We need the confirmation, yes, that our plasmid or our gene of interest is present over here. Our gene of interest is present over here. So when you will grow them on culture with XGAL and IPTG, they are going to give you the white colonies. The white colonies are the recombinant one. Okay, so that's why, but the blue colonies are not the recombinant ones. That's why this screening is termed as blue-white screening. So there are different methods also, but this method is widely acceptable in the biotechnology. Thank you.